Okay, this is a presentation on cancer genetics, and this is just general cancer genetics, but we're going to discuss a few disorders in here. So, cancer prevalence, it's estimated that 1 out of every 3 individuals will develop cancer in their lifetime, which is a lot. This is actually from 2018. I couldn't find the 2019 one, but they actually haven't changed a lot recently. In males, the most common form of new cancer is prostate. The most common form of diagnosed cancer in females is breast. However, as far as cancer deaths, the most common in both men and women is lung. So while prostate and breast are the most common diagnosed, a lot of people survive that. Whereas, unfortunately, lung cancer, which is preventable in many cases by stopping smoking, is the most common cause of cancer death in both men and women. So I'm just going to go through a few definitions. One thing I want to say is these are pretty basic definitions, but if there's any semantics that are different with pathology, please go with pathology. So a neoplasia is a disease process characterized by uncontrolled cellular proliferation leading to a mass or tumor. Uncontrolled or unregulated cell growth resulting in a mass, and not all neoplasias are cancerous. Malignant is uncontrolled cell growth that is capable of invading other surrounding tissues, and that's really the definition of malignant. It can travel to other sites through the lymph nodes. Metastasize is when the neoplasia spreads to the distant sites, for instance, breast cancer metastasizing to the liver. And a benign tumor is a tumor neoplasia that does not invade or metastasize other tissues, but it still can be a problem, especially if it's in a limited space, such as a meningioma in the brain. It's a benign tumor. It's not going to spread to other places, but it can cause a lot of problems because the brain is a, encased organ. Sarcoma, whenever I hear the word sarcoma, it scares me as a tumor. Forms from meningeal muscle, bone, connective tissue, or nervous tissue. Carcinoma forms from epithelial cells, linings of organs such as the breast and the intestines, hematopoietic and lymphoid malignancies. And we're going to discuss this in more detail when we get to oncology. And it spreads through the blood and bone marrow and lymphatic system, so leukemias and lymphomas. The most important thing to understand when talking about cancers, and these things change on a regular basis, but if you understand the concept of the cell cycle, as you move forward in medicine and as new things come up, you're going to understand it better. So the cell cycle begins with mitosis. The first cell stage is G1, and this is when the cell is growing. And then it has to decide if it's going to divide. So S is when we replicate our 22 autosomes, our sex chromosomes, our 22,000 genes. And the G2 is super important because did we do this right? Even if we're 99.99% right, that's still a ton of mistakes. Cancer cells like to skip G2. They like lots of mistakes, and they like to keep propagating more and more and more mistakes. And this leads to uncontrolled growth and lots of damage to the DNA. So, as I said, there's checkpoints to this. There's the cell, if you're in G0 and there's not enough nutrients, or the cell's not large enough, or there's a lot of DNA damage, the cell decides you're not going to go and replicate and divide again. You're just going to go into a resting state. You're going to stop growing. That's G0. And then there's another stage, and so that's in the G1 checkpoint. So there's another stage at the end of G2. Did you make enough? Did you fix all the DNA errors that you made when you were copying your DNA? Because we do have a mechanism to do that. Did that happen? And if it didn't, nope, you're not dividing. And then there's also a metaphase checkpoint to make sure the spindle formed correctly and everything was going right there. So this is actually a really important slide to understand because cancer is really the balance between proliferation, growth, and mitosis, and attrition, cell death, and apoptosis. So there are a lot of things that can change this balance. UV radiation, viruses, ionizing radiation, medications, tobacco, chemicals, and really the normal cell process. And then finally, heredity. And so, for instance, we know that tobacco causes damage to the lungs. And the problem with that damage is whenever a cell is damaged, it has to divide more than normal. Whenever a cell has to divide more than normal, something bad can happen, a mistake can happen. We will probably learn in 30, 40 years that these kids that are vaping and damaging their lungs will also be at increased risk of lung cancer because we know that they've damaged their lungs. And it doesn't happen today or tomorrow. It doesn't happen when they're 20. It happens over time. Radiation. Radiation affects a lot of cells. And when we first, they used to have, in shoe stores, they used to check if your shoes fit by putting your, feet into a x-ray machine. And then a lot of people, got cancer from that. So, we're learning about these things on a daily basis. This is another really important concept, and that is all cancer is genetic, but not all cancer is hereditary. So if I look at a cancer cell, I'm likely to see a lot of damage. But that doesn't necessarily mean that the person was born with that damage. Oftentimes, it's an accumulation of damage over time. So let's just look at this. Here's a bunch of cells, and they're all normal for a number of different genes that control cell growth. And then we have a mutation of one of the genes, and so we have a bunch of cells with one mutation. But we're okay because we still have a lot of other genes that are working. And then we lose a second gene. And then we lose a third gene. And in a small group of cells, we now have 4 different mutations of genes that control the cell cycle. And now this cell is no longer controlled, and it starts replicating very quickly and can take over this whole population. This is not hereditary. This is sporadic. This happened because of an accumulation of mutations, and that's why often cancer is seen later in life. It takes a while to accumulate all those mutations. So tumor suppressor genes, the function of these genes is to constrain growth, is to keep growth just right, not too fast, not too slow, just right. The loss of these mutations in one of the 2 copies in and of itself does not cause cancer. The loss of function in both can result in loss of control of growth. So here we have 2 copies of the tumor suppressor gene. They're both working just fine. In a small population, we lose one, and we're still okay. We still have normal growth, but if we lose that second copy, that's a second mutation, now we've lost control and we have uncontrolled cell growth. The vast majority of hereditary cancers are caused by being born with 1 copy. And again, you have normal cell growth unless you lose the second copy. And that's known as the Knudsen 2-hit hypothesis. So in the Knudsen 2-hit hypothesis, here we have 2 normal, you're born with 2 normal, let's say tumor suppressor genes. In a group of cells, you lose 1, but you're still okay, but then you lose the second, and you have uncontrolled cell growth. But what if you're born with 1 copy of this gene that's not functioning? Every cell in your body, every skin cell, every liver cell, every brain cell has a copy that's not working. Now it only takes 1 event, not 2, to get yourself into trouble. Now, having said all that about hereditary cancer, the vast majority of cancer is sporadic. And again, you're going to learn about this, and they're looking at this all the time now. They're looking at tumor cells. They're finding all kinds of mutations. But the question is, that we expect. That's why you have cancer. But was that inherited? or was that sporadic? So the vast majority of cancer is sporadic. Then about 10 to 20% is familial. And this little teeny sliver, only 5 to 10% is hereditary. But we're now going to talk about the hereditary cancer of breast. So sporadic versus familial. Sporadic results from a buildup of damage in the cells over time. Usually we see cancer at an older age, usually older than 50. More common cancers, like, for instance, women with breast cancer, and no significant other family members with a similar cancer. Familial, on the other hand, is a combination of genetic and environmental and lifestyle, not a single inherited gene. And there seems like there's more affected people in the family, but, than would be expected. But sometimes I'll take a family history and there's 3 or 4 individuals with lung cancer, but everyone smoked. And they were probably, even as children, exposed to smoke. And then you get to hereditary, results from inherited gene mutations that predispose a person to develop cancer. Some hereditary cancer syndromes can result from de novo mutation. And what that means is the individual has a cancer that's hereditary, but we don't necessarily see a family history of it because it was a new mutation in the germline of that individual and when the egg or sperm was formed. Mutations don't actually skip generations, but the cancer can. And there's over 100 different genes that we know of today that can increase the risk of cancer. And the key here is that we know of today, because when I went to medical school, I don't think we knew about any of them, and then we knew about the breast cancer 1 and 2 genes. And now there's hundreds. And so there'll probably be thousands within your, practice of medicine. But I want to talk about a couple super rare genetic mutations that can result in cancer. And one is retinoblastoma. And this is a malignant tumor of the eye. It's incredibly rare. But this is why we like to look at the red reflex. Here you can see this kind of pops out white. It's caused by the RB1 gene that's located on long arm Q of chromosome 14. And it's a tumor suppressor gene that affects G1. It affects the cells being able to replicate. Like, it makes sure that there's not too much damage. It's autosomal dominant and highly penetrant. 40% of hereditary. So when we see a retinoblastoma, there's a 40% chance that there's a germline mutation. And when there is a germline mutation of this gene, the individual is at a higher risk of bilateral retinoblastoma. So even though one eye is affected, they're at a significant risk of the other eye becoming affected. The inherited cancer, no inherited cancer scares me more than Li-Fraumeni. Li-Fraumeni is a mutation or deletion of this gene are often seen when we're looking at tumors. For instance, most tumors have some type of either deletion of this gene or mutation of this gene when we actually look at the tumor cells. But when you're born with this, that's what Li-Fraumeni cell is. And Li-Fraumeni is a disorder when the individual is born with a mutation of this gene, TP53, or some people call it P53, which is located on the short arm of chromosome 17. And this one is a really strong tumor suppressor gene that affects both G1 and G2. Fortunately, it's an incredibly rare disorder as far as being familial. But when you look at the family history, it's to me surprising that these people could even reproduce. So here's an individual. Here's our patient. She's 35 with breast cancer, which is pretty young for breast cancer, and she has 2 kids. One daughter got a sarcoma at 7, and another daughter got leukemia at 6. In general, we're going to talk about leukemias in the cancer section. That's not necessarily inherited, but when you start looking at the rest of this family history, you start questioning in this family. But sarcoma, again, is a scary word. And this woman, her father is 50 and he doesn't have cancer, but his sister had breast cancer at 40 and bone cancer, osteosarcoma at 42, and his brother died at 33 of leukemia, and his brother's son died at 32 from a brain tumor. and her paternal grandmother died at 40 of bilateral breast cancer. That's a lot of cancers, a lot of different cancers. And just one more FYI about Lee Fermini. I talked to one of the students that just took step 1, and there was actually a pedigree of Lee Fermini. So Lee Fermini, you see a pedigree with every possible horrible cancer you could possibly imagine at super young ages. Think Lee Fermini. So, tumor suppressor genes, the G2, again, Lee Fermini, the TP53, and the BRCA1 and 2, which we're going to talk about in life cycles 2, and then the G1, the retinoblastoma, and the Lee Fermini as well. DNA repair. DNA makes mistakes, and they need to repair it efficiently. So the G2 checkpoint makes sure it was done. But DNA repair is the process of repairing it. And there are some inherited disorders of DNA repair, including xeroderma pigmentosa and ataxia telangiectasia. These are super rare disorders. They don't really fit anywhere, so that's why I'm putting him here. So when you start studying for step 1, you'll have something to back you up here. So xeroderma pigmentosa is caused by mutation of the gene involved in DNA repair at G1. And the mutation of this gene prevents apoptosis when the DNA is too damaged. So it prevents it from going into the into cell division. This is inherited in autosomal recessive fashion. Both copies of the gene are non-functional, and the patients are sensitive to UV radiation, high frequency of skin cancers, including squamous cell, basal cell, and melanoma. These patients should really not be out in the sun during the day. And again, like I said, autosomal recessive. Most inherited cancers are dominant. Both copies of the gene from both mom and dad are non-functional before the individual is affected with this. Ataxia telangiectasia is another recessive and a defect in DNA repair in both S and G2. It's caused by the ATM gene, and features include telangiectasias of the eye, cerebellar ataxia, and immunodeficiency. So you might have reviewed this a little bit in the immunodeficiency case. So hereditary cancers, clues in the personal and family history, young age of diagnosis. Individuals under the age of 50 that are diagnosed with cancer. Multiple blood relatives on the same side of the family with similar types of cancers. And I'll give you an example. We see a patient, they have breast cancer at 55, and their mom had breast cancer at 75, and their father's mother had breast cancer at 72. That's not, yes, there's 2 members of her family that had breast cancer, but they were both older, and they were from her mom's side and her dad's, and they were not from the same side. So it wouldn't, I'd be more concerned if this woman that had breast cancer at 55 had a mother who had breast cancer at 50, and her sister had either breast or ovarian cancer at a young age. Then that would be more concerning than older individuals on both sides. Consider other risk factors, like I said, multiple lung cancers and smokers. Whenever I'm taking family history and somebody says my relative had lung cancer, the next question I always ask is, were they a smoker? Because there are some more concerning types of lung cancers in non-smokers. But if they're a smoker, and we might see many members in a similar family that had lung cancer when they're smokers. Individuals with multiple primary cancers, they have 2 different types of cancers, and related cancers in the family. And we're going to talk about breast and ovarian, rare type of cancers like males that develop breast cancer. So in conclusion, if you look through the chromosomes and genes of malignant tumors, they're going to be abnormal. You're going to see gene mutations. But in most cases, it happened in those cells as an accumulation that ended up resulting in the tumor because of loss of regulation. Most inherited cancers are dominant. The second copy is lost in a portion of the cells, And that's why not everybody with these inherited cancer genes actually develop cancer. Xeroderma pigmentosa and ATC telangiectasia are inherited in the autosomal recessive fashion.